as I've said, let me see if I can pass my slides. Yeah. My main interest is in understanding tropical rainforest evolution. Here you have a drone shot from the Yasuni National Park in Amazonian Ecuador in South America. And it's been labeled as one, if not the most biodiverse area on the planet, at least based on tree species. There are about 300 different tree species cramped into a small area, not larger than a soccer field and a half. And so trying to understand the evolution of these ecosystems is very challenging, as it's highly complex and a lot of interactions and a lot of different ways we can try and understand how biodiversity has evolved in these areas. But rainforests are also one of the most threatened ecosystems on the planet. And so our data also serves as a fundamental basis to try and understand how we can better conserve this very important biodiversity, not just for local communities who use and need these forests on a daily basis, but also at a global scale, as rainforests are very important for global climate mitigation in general. So I would qualify myself as a botanist, and I'm interested in biodiversity and evolution, and I try to answer two main questions: what are rainforests made of? How can we identify the different species that occur in rainforests, and how has this biodiversity evolved through time? And I do a lot of field work in rainforests across the different rainforest areas. And I have two main interests in terms of taxonomy, and these are palms, which many of you must know, and another major tropical plant family called Annonaceae, which is less known, especially in northern areas, but in South America or in Southeast Asia, Annonaceae is very, very important. They give different types of fruits that you can eat. And one species in Southeast Asia is also the basis of the perfume of Coco Chanel, which is a very well-known perfume. My slides to change. So today I'm going to talk to you about three major projects that I've led over the past few years. The first one is going to be concentrating at a regional level in Africa. Another one is this ongoing project that I'm leading, which has to do with, which is at a global scale. And then we're going to zoom in into a conservation project I'm participating in Western Ecuador. So the first stop here in our trip is Africa. And Africa is really a land of contrasts. And as you can see here on this satellite image, to the north, we have the largest desert in the world, the Sahara, which is also probably the least biodiverse area in terms of plant species in the world. To the extreme south of Africa, we have the fine bios ecosystem, which has one of the highest concentration of endemic plant species in the world. And in the middle, we have different types of ecosystems, which are very important. And in particular, the tropical rainforests of Africa, which extend from West Africa all the way to East Africa, and passing by the Congo Basin, which is the second largest continuous extent of tropical rainforest after the Amazon Basin. In terms of plant diversity, Africa has always been labeled as an odd man out, a term that dates back to Richards in 1973. And that is because Africa has shown that there are generally less diversity of seed plants occurring in Africa than compared to the neotropics or to Southeast Asia. And this has puzzled biogeographers for many years. But even so, Africa is a very diverse continent, and especially the tropical rainforest. For example, in the 50-hectare plot of the Korup National Park in Western Cameroon, they've measured they've counted about 130 tree species occurring there. So even though we talk about an odd man out, it's still extremely diverse. And it's a wrong label to say that Africa is species poor. It's actually very species rich, but when compared to other regions, these other regions are even more species rich. Africa, in fact, has a very unique geography. The African continent sits in the middle of a plate, So it doesn't occur at the junction of different plates like in South America or in Southeast Asia. And so the tectonics of the continent are very passive. The most important tectonic activity is the origin of the East African Rift Valley, which is world known for being potentially the birth sites of humans. And you can see this here in red and in white indicate these very high elevations. In fact, the topology of Africa is bimodal. We have the west central part, which is very low, here characterized in green, and then the east southern part of Africa, which is very highly elevated. And on average, compared to the neotropics, for example, Africa is actually higher in elevation on average, even though the neotropics have the Andes. So Africa has this unique setting. It's also you can also characterize this by it not having that many earthquakes, especially in the areas where rainforests occur. And Africa, as like any area on the planet, has suffered through many different major geoclimatic periods. This was summarized in a paper we published recently in 2001 about the evolution of biodiversity in Africa. And we identified in this publication 6 major geoclimatic periods which could have impacted tropical diversity in Africa. One example is and these events have led to the origin of different biodiversity events in animals as well as in plants. And so when we study these across time, we can find that many of the origin of these species or genera or clades date back to important geoclimatic events that Africa has known. One example that we recently published was this idea of sequential diversification. And so one of the ideas in Africa is that aridification has had generally a negative impact. In this paper that we published, 2 weeks ago, my PhD student, Léopold Dagalier, showed that actually we do find an increase in extinction rates, which is here characterized by these blue dash lines. But this was immediately followed by an increase in speciation rates. And this we suggested this could be a sequential diversification scenario where we have extinction, but then it was followed by opportunities linked to creation of new biomes or vicariance events between different areas which enhance speciation. So in this talk about Africa, I first wanted to talk about biodiversity, because amongst all continents, the study of biodiversity, of biome limitations and of flora, of the flora of Africa, has had a very long history, more so than in any other continent. And very early on, Frank White published a map of the delimitations of the different biomes, which he called phytochoria. And you can see a map here where he shows all these different biomes. And what is interesting with Frank White is that this was mainly based on his expert knowledge of the continent. It wasn't really based on any kind of underlining hardcore data. Several years later, Peter Linder and his research lab started to use the distribution of species to try and delimit and understand biodiversity, plant biodiversity distribution in Africa. And in this case, they used a database of about 5,000 species distributed across tropical Africa. And finally, many people have been using GBIF, which is a large repository of plant distribution and animal distribution data. And they were also able to use this information to understand the distribution of biodiversity, of plant biodiversity in Africa. But overall, these data sets were either based on few expert or unchecked data. GBIF has a lot of data, but most of the time it's unchecked, it's unverified by taxonomic experts. So in this Rainbow project, we set out to try and understand the distribution of plant biodiversity using herbarium data and we synthesized about 13 databases and 8,000 plant specimens collected in tropical Africa. We collaborated with numerous botanists across our study area, as well as numerous herbaria. We undertook several multiple quality checks to try and make sure that our database was really standardized and taxonomically verified. And after several months, this led to the generation of a data set which is now a bit old, it's 6 years old, which we called Rainbow. And so this database documents about 600 georeference unique herbarium specimens across the continent and documents the distribution of about 25,000 plant species across this area. You can see here the total number of specimens per grid cell, and you can see that it's pretty heterogeneous, with some areas like here in Gabon and in Cameroon being pretty well covered, whereas some others like Central Congo have yet little collections and need to be more botanically explored. When we have a database like this, we can analyze many different things. And one of the first analysis we tried to do is to try and estimate how many plant species there were per country. It's a very fundamental number that every country should have, is how many plant species do I have? Most of the countries in the north have easy access to that number because they have a lot of research around that, but also because the flora isn't that diverse. But in some countries, this type of data is really hard to come by. So we use different types of statistics, and we've shown, for example, that 3 countries stand out as having way more diversity, plant diversity than the others. These are Cameroon, Tanzania, and the Democratic Republic of Congo. The Democratic Republic of Congo is the largest country in our study area. That could explain the high level of diversity. Cameroon and Tanzania, in contrast, are smaller, but they're really at biogeographic crossroads. Cameroon, for example, has an influence from West Africa, Central Africa, and also South Africa, which could suggest or could explain these higher levels of diversity. We can also estimate, for example, the total number of tree species. So several years ago, people working in the Amazon basin came up with a suggestion that there were about just under 12,000 tree species documented in the Amazon basin. So we tried to do the same with our database, and we came up with a much small number, 3,000 tree species documented in the tropical rainforests of Africa, here in these different shades of green. And this shows once again that we have 4 times less tree species documented in tropical Africa than for the Amazon basin. But nevertheless, most of these trees were endemic to the region. But on the other hand, it could be that African tree diversity is underestimated, especially widespread tree species, which are named but are very little investigated. And when we look closer to these widespread tree species, we see that in fact we have a lot of cryptic diversity. We can also use this database to try and understand the limits between different bioregions in Africa, following in the footprints of Frank White that I introduced a bit earlier. And so here, using a very large database of 25,000 plant species, we undertook a network analysis, and we documented 16 different bioregions and 12 transition zones. And what's really interesting is that even though we have all this data and these new ways of analyzing, they confirm relatively well the expert knowledge of Frank White, which is an interesting result that we had. And this data set is now used, or this result is now used as a more accurate way to delimit these different regions across Africa. So the other point about these is that, okay, this is all very good, but how can we inform conservation with these types of data sets? Don't know if anybody in the room knows, but the IUCN produces a list, which is called the Red List, which is one of the most important lists, which informs the threat level of a species. The IUCN Red List is a accepted and authoritative list. It's actually used now as one of the gold standards to understand or to lead different international agreements on biodiversity, like during the COP15. Basically, if I can summarize the importance of the IUCN Red List by one of my colleagues, who said that a species doesn't exist if it's not on the red list. And that's really true for companies, for governments. If it's not on the red list, companies don't have to take that into account when they do different kinds of environmental planning throughout their activities. Unfortunately for plants, this red list has less than 7% of species documented on it. So there's a really important need to accelerate this process. But when we accelerate the assessment of threat, we always need to follow this IUCN methodology because of the reasons that I explained just before. Because if we don't do it that way, then it won't end up on the red list, and then it won't have any kind of impact in conservation. And so in this project Rainbow, we set out to try and generate an R-based package that will allow you to generate conservation assessments really fast once you have the distribution data. I just want to really underline this word preliminary. This CONAR program would be able to generate preliminary conservation assessments, but they always need to be thoroughly examined by experts of the region before they can be validated. But at least it moves the assessments fast, and it generates all the data that you need to be able to do a full conservation assessment. So using our Rainbow database, we undertook a CONAR analysis. So here we're talking about a very large scale continental-wide analysis. So we can't do this on a species by species basis. But the analysis of 22,000 species took about 6 hours. And we found after numerous rounds and checking that about 1/3 of the tropical flora of Africa was threatened by extinction, which is a relatively large number, and which provides a lot of insight into how this flora is threatened. We can also look at the distribution of threats spatially. So here in this map, we underlined levels of high proportion of threatened species. So compared to the total flora, how many species are threatened? And so the more bluer colors represents area where there's a high level of threat to the flora in general. And we highlighted two main areas. The first one are the Ethiopian highlands, which are an area which have a very long history of human occupation and human agriculture especially. So a lot of the original vegetation has disappeared and replaced by different types of agriculture. And West Africa has undergone a very high level of deforestation rates throughout the last decades. And that is why we have these high levels of threat. A lot of the species there are threatened. In contrast, we found areas where we have very little or lower proportions of threatened species. And this, for example, is in the area of the Congo Basin, Cameroon, and Gabon. In these areas, large expanses of rainforest still exist, and deforestation hasn't been as intensive as in West Africa, although deforestation nowadays is increasing. And so we really have to keep an eye on these regions so that we don't end up in a similar situation as in West Africa. Once again, we said that there were 32% of species that are threatened, but this is mainly taxon dependent. For example, a very important genus, Coffea, of which we have the coffee bean, has about 60% of its species threatened. In contrast to a group like palms in Africa, although there are very few species, only 10% of these species are threatened with extinction. So it's really also a group-dependent analysis, but overall, about 32% of species have been identified as potentially threatened with extinction. So finally, in this section, another thing we can do is try to understand the evolution of biodiversity in the area, once again using this Rainbow database. One thing that we can do when we include phylogenies, especially dated phylogenies. So a dated phylogeny, you have this on the left-hand side of the screen, represents the phylogenetic relationships between species calibrated in function of time. So we can use this phylogeny to identify recently evolved species and species that have been around, or at least species that occur on lineages that have been around for a long time. And if we fuse that with the distribution, we can identify two types of endemics. We have neoendemics, which are species which have a very small distribution and are very recent. And then we have paleoendemics, which have a small distribution, but are very old, sitting on very long evolutionary branches. And if we do diversity analysis, we can find areas which have a high concentration of neoendemics. And this would translate the idea that that region somehow is a cradle of diversity. A lot of new species are being born there. And in contrast, if we have a high level of paleoendemics in a region, this translates to the idea of persistence or of museum of diversity, where in fact something there allows the persistence of these species through time because they sit on these very long evolutionary branches. And in Africa, there were several hypotheses that were emitted about trying to understand where these cradles or museums of diversity occurred. Some suggested that mountains were cradles of diversity because mountains are very heterogeneous. There are a lot of uplift going on very fast. So this was probably a very good place to see the birth of many different recent species. Other people suggested that mountains were actually museums and cradles. Although, as I said before, there's a lot of heterogeneity. mountains also have these gradients, and so species can escape climate change very quickly just by moving very short periods of short distances up or down the mountain. So they would also be areas where diversity would be able to persist through time. And finally, we also have these kind of contrasting ideas about lowland rainforests, which are either museums of diversity. Rainforests are very stable ecosystems through time, so specimens or species have accumulated through time. Or in contrast, rainforests are very dynamic ecosystems, very complex, a lot of interactions which gave rise to many different species through time, and they were labeled as more like cradles of diversity. So in this context, we used the Rainbow database to try and identify areas of neo, paleo, or mixed diversity. We used the Rainbow database. We used the recently generated dated phylogeny, which included 1,700 genera. And we fused these two approaches together in a program called Canopy, Categorical Analysis of Neo and Paleo Endemism, which allows us to identify exactly that, areas of high concentration of neo-endemics, paleo-endemics, and mixed endemicity. Mixed endemicity is an area where we have both a high concentration of neo and paleo-endemics. When we applied this to our database across tropical Africa, what we found is that we had very few areas which corresponded either to a neo-endemics or paleo-endemics, but we had high concentration of areas which were mixed endemics. And as you can see here in blue, these occur along the East African Rift Valley, along these mountainous areas in tropical Africa. And so this kind of confirmed the idea that the East African mountains are probably areas of mixed endemicity, that they act both as museums of diversity as well as cradles of diversity. And we found that these areas of mixed diversity were correlated with altitude. As we go up, they become we have more areas of mixed diversity. We also found interesting signals that the lowland Guinea, which is the Congo Basin, basically mainly acts as a museum of diversity, especially at the genus level. So now I'm going to transit from this regional level into looking at global approaches. So throughout my career, I've been focusing on Africa. In a previous project, I was focusing on the neotropics. But I came up with this idea that we should probably look at rainforests at a global scale. And so we were lucky enough to get this big funding from the European Commission, which funded this project. And the idea was to try and understand rainforest evolution, but taking this very global view and being able to compare also regions between each other. And so basically here, we're trying to understand the evolution of rainforests across the world, trying to understand the evolutionary processes that define rainforest evolution, but also in a second phase of the project to try and understand the impact of this ongoing 6 mass extinction that we're witnessing during this era that we call the Anthropocene. How will extinction impact diversity of rainforests? And so the literature on understanding rainforest diversification is very long, very old, and relatively complex. But we can try and, if we simplify this to the extreme, there were 2 main hypotheses to explain diversification, as I already introduced before. The museum hypothesis, which suggested that rainforest witnessed very low extinction because this is a very ancient biome that's always been around for about more than 100 million years. In contrast, and this was something that we documented in palms, for example, where we saw that lineages of palms seem to increase on a regular basis within this biome. And the other alternative hypothesis was that, which is not mutually exclusive, is that rainforests are actually cradles of diversity, very complex, lots of interactions, which led to high diversification. And this was shown in typical genera of the neotropics, for example, in Inga first, and also in an Ananaceae genus called Guaiteria, where a lot of this diversity, they're very hyper-diverse genera with more than 100 species each. A lot of this diversity originated about 5 million years ago. Another way to look at rainforest evolution is to look at biotic versus abiotic interactions. So once again, I also hinted toward this in my introduction, but abiotic events linked to climate change, tectonics, were suggested to play a very important role in the evolution of rainforests. One major example is the uplist of the Andes in the neotropics, which was suggested to trigger diversification in a diverse family such as Rubiaceae, the coffee family, but also at generic levels. For example, a diversification of Lupinus, which is a very important lineage in the Andes. In opposite, biotic interactions have to do with pollination syndromes, dispersal events of innovations in dispersal, but also a major important biotic interaction is herbivory. Tropical rainforest is an ecosystem where it's hot and humid all year round, and so it's beaming with life, and generally it's beaming with a lot of insects, and insects like to eat leaves. And so plants in that area have a very strong herbivory pressure. And so Elrich and Raven came up with this hypothesis, amongst others, that the high level of species diversity in rainforest could be linked to this sort of arms race between plants trying to escape being eaten by insects, and insects trying to catch up. And one way for plants to escape herbivory, besides evolving hairs, all kinds of defense mechanisms, is chemical defense. So they evolve these very unappetizing leaves for insects, and they can escape this pressure. This is something that was also shown in Inga, led by one of my colleagues, Maria Jose Endara in Ecuador, which have these incredible, fascinating studies about how Inga has been able to escape herbivory pressure by evolving all kinds of different traits, including chemical traits. Another part of the project is to understand regional versus local evolution, in particular to try and address this odd man out pattern of evolution. So when we put all this together, the project tries to understand diversification, biotic versus abiotic, regional scale processes, but also, as I said in the last part, trying to look at ongoing extinction. Now, the project is midway through. We're going to finish in 2026. So I don't have much to present, but I'll show you some advances, and in particular, the results of a PhD student that we had. The idea of the project is to really try and generate, one way to study rainforest evolution is to reconstruct molecular phylogenetics. And so what we wanted to do in this project was to reconstruct for the first time a complete phylogeny for a very important pan-tropical plant family. And what this does, it allows us to go from shallow levels, so really deep evolutionary questions, all the way to the much more recent evolution of plants. And in doing so, we can address different levels and different scales of hypothesis where these actions probably are acting. For example, we know that the cradle hypothesis and the Red Queen hypothesis of this arms race generally occur at more recent timeframes, whereas the global abiotic impacts in the museum model probably occur at much larger scales. So by having a very densely sampled phylogeny, we can generally address these questions more in detail. And so for the project, we also wanted to generate novel and complete data at global scales for all species in a given clade. And to do that is not going to be a surprise to you, but we're going to do this in Annonaceae. And so Annonaceae is proposed as a model for tropical rainforest evolution, not because it's better than any other tropical rainforest, but because it has a number of attributes that allows us to undertake such a project in a limited time frame, which was 5 years. So first, Annonaceae is one of the top 10 most biodiverse families in tropical rainforests. It's a pretty large family with about 2,500, 2,400 extant species. So it's diverse, but it's not too diverse. We're not talking about Orchidaceae where we have 20,000 species. So we can reasonably expect to get information for all species in a reasonably amount of time. It's morphologically diverse. You can see here examples of some of the flowers and some of the fruits. It's also interesting because it has a very diverse plant chemistry. So that's really nice. We can test all kinds of hypotheses. And one of the most important parts is that it evolved about 90 million years ago. So that's really at the start of the modern tropical rainforest evolution. And the most important part of all is that we have a very solid taxonomic background. And this has been built over decades of studying this family in Holland in particular, but in other areas too. And so taxonomy is really the basis of this project. Why? Because we're not going to lose or waste any time trying to understand species limits or fall into taxonomic questions about, oh, but what is this species and how can we identify? Most Ananaceae genera have been revised recently, so it allows us to move forward pretty fast. The other star of this project is the herbarium specimen. Everything we do revolves about using herbarium specimens. We can get morphology, we can get DNA, I'll come back to that a bit later on. We can get distribution, and more recently, we can also use technologies such as near-infrared scanning to even extract the different types of chemicals that these herbarium specimens have in their leaves, which allows us to address all kinds of interesting hypotheses too. So for the first part, we have a global collaboration with people everywhere, in particular people living in the tropics, which are principally concerned by this project in South America and Africa and Madagascar and India and in Southeast Asia. And we depend on different herbaria, in particular the herbarium in Paris and the herbarium in Naturalis, because they're historically are the largest museums of Ananaceae collections. We hired a technician called Carlos, an engineer, who spent 2-3 years going around different herbaria and sampling these herbarium specimens. It's a very tedious and long job, but he did that very thoroughly. These are then sent back to our lab here in Montpellier, and this was the work of Vincent Soulet, who extracted about 5,000 herbarium specimens and undertook the sequencing of nearly 3,000 individuals. All this led to, we were able to achieve the sequencing of 2,050 species. That's about 84% of the diversity. And the oldest specimen that we sequenced was collected back in 1785. I just wanted to dwell a bit on that. This was collected by a very famous expedition by two Spanish botanists, Ruiz and Pabon, who went to Peru for the first time, collecting, documenting the diversity of that country. Well, at the time it wasn't Peru per se, they collected this specimen. This is the actual specimen that we sampled, and for which we extracted and sequenced DNA. So it's always a fascinating thought to see how people who participated in the expedition and witnessed the collection of this specimen, how they would react nowadays if we told them, well, 200 years later, we were able. If I were to go back in time and talk to Luis and say, look, you're making this collection, and in 300 years, we'll be able to extract DNA, use near-infrared scanning, I think his mind would have been blown at the time. So using all this huge effort, and this is a view of most of the people working on the project, we were able to sequence 2,000 species, and this is the phylogeny that we got. So this is one of the biggest phylogenies of a tropical plant family ever to be generated to date. And this is mainly thanks to all the effort of all these colleagues of mine and who work for the project. One project that has been ongoing for some time is the PhD project of Paula Santa Cruz. She's an Ecuadorian student, and she had this incredible task of trying to understand interactions between herbivory and Ananaceae. Before her PhD, we knew almost nothing about how Ananaceae defended themselves or reacted to herbivores, or even what herbivores would eat these plants. And so Paula spent a year, every month for a year, a week, during every month for a whole year in Yasuni, trying to understand the interactions of 10 Ananaceae species. And what's interesting in Yasuni is that we have representatives of all the major subfamilies of Ananaceae. So in Yasuni, we have about 90 million years of evolution going on, and allowed us to get a very broad overview of the diversity. This was done in collaboration with Maria Jose Endara from the UDLA University, and she's one of the foremost experts in understanding plant herbivore interactions in the tropics. And so here we can see Paula and Maria Jose in Yasuni documenting for the first time these interactions. Overall, after this year, we found a discrete but diverse herbivore community. It was very hard to find these herbivores. I remember the time when Paula found her first herbivore. She was very, very happy. It was a very good moment because it took us a long time. But after a year working there, we're able to document all these wonderful insects. Most of these are obviously butterfly larvae. The next steps of her project is to document this. We found an incredible amount of new interactions that were not described for NNAC, obviously, but she's also going to go further looking at the secondary metabolites, so all these secondary chemistries that potentially are involved in trying to understand herbivore interactions, and in particular to understand if different species have different profiles, secondary metabolite profiles or not, and how that evolved throughout the tree of life in Ananaceae. So one way to also understand this is to look at near-infrared scanning. Near-infrared scanning basically looks at the vibration of molecules and produces a spectra. And so once again, Carlos Rodriguez scanned thousands of herbarium specimens to try and get this type of information. Here is what it looks like. We take the herbarium specimen. It's non-destructive. We don't have to destroy the herbarium specimen. And then we can generate these plots, as you can see here. The first analysis of this NIRS data on Ananaceae was done by a master's student here in Montpellier, an American student called Lily Bennett, and she did a wonderful job at trying to untangle all these spectra and to plot them on phylogenies. And this was done for the first time in our lab, and it gave these really interesting results that I won't have too much time to discuss here at this point. I'm going to go quick through these things, but we're looking at different types of distribution patterns, and for that we need to generate a database of distribution, and we're using citizen science to do that. So in this case, citizens have access to herbarium specimens, and then they can log different types of information like georeference the specimens. This was done, for example, for Madagascar. Here we have the distribution of Malagasy Ananaceae, and it was the study of my master's student, Erika Ravo-Manan, and picked up now by PhD student Serafin, who's working on the distribution of Ananaceae at global scales. Finally, we're also looking at paleoclimatic modeling. This is a very exciting project led by Pierre Sepulcre and implicating my postdoc student Delphine Tardif, where we're trying to look and model paleoclimatic through the Cenozoic, so the last 100 million years. But instead of having one estimate of climate, we're having what we call a discretized or a spatialized paleoclimatic across the globe. This means that we can point basically at any area on the globe, and we can have an information about how the paleoclimatic evolved through time. And again, I could talk a lot about this, but I have to move on to the next part of my talk because I think I'm running a bit out of time. Last but not least in this project, always important to say that taxonomy is always the basis of what we do. And we were able to, thanks to this grant, finish the flora of Cameroon, which is really a very long project that I started over 15 years ago, in which crystallized in this publication where we describe about 165 Ananaceae species for the country, together with many, many students and colleagues from Cameroon. So the last part of my talk, which is really new for me, comes from this idea of thinking globally but acting locally. And some years ago, I read this publication by Bill Lawrence, one of the foremost tropical ecologists, and he talked about adopting a forest. And he basically says that my advice to aspiring conservationists, so hopefully several of you in the room, be it in the tropics or elsewhere, is to find yourself an imperiled patch on Earth, anywhere on Earth, tend to it, defend it, and make it your own, if only in spirit. Forge alliances, build bridges, encourage others to share your vision. Do not expect it to be easy, it will not be. But by adopting a forest, you might just create a lasting legacy. I'm not really interested in the lasting legacy, but I was interested in trying to adopt a forest and really help people conserve that forest through time, local people. And I've been wandering around rainforests a lot, and I found this forest in the western Choco or the Choco of Ecuador, which is this western area, this western rainforest in Ecuador. Unfortunately, the Choco of Ecuador is also known for being one of the areas where we had a very long, uncontrolled deforestation. And so here you can see the Choco rainforest as it was before and how it went and being deforested through time. And so now we only end up with very small patches. And this is where this patch of rainforest occurs. But the most important person in this project or in this quest is Agnello Lor. Agnello Lor is what we call a parataxonomist. He's He's somebody who worked for universities, and he has this incredible love for tropical rainforests. And he's a native of that area. And several years ago, he bought some land on which there was a tropical rainforest of about 7 hectares. And he's really a conservation hero. He's not somebody with a lot of money. He has a very humble lifestyle, but he gives everything he has to protect and document that rainforest. Even if he doesn't have much recourse, he's going to give everything he has. And so here is a satellite view of where this area is. This is, I don't know if you can see my mouse, I hope so. But this is in the middle of the screen. You can see the rainforest that we were addressing, and all around the rainforest is just pasture, pasture, pasture. Any kind of forest that you can see here are along very steep ravines along rivers. So this is one of the last remaining patches of the area. This is kind of what it looks. So here in the left photo, you can see a bit of the rainforest that we're protecting, and then you can see that all around it's just basically pastures for cow and other types of crops. And on the right, you can see an endemic palm called Atalea colenda, which was left standing on top of this hill that was completely shaved just so that a bunch of cows can eat. Here you have a view of the imperiled patch. On the left side, top left side of your screen, you have the research station that Anelio built from scratch all by himself. Obviously, with the help of people, but he funded this completely by himself, which is really a testimony to his passion and dedication that he had. This is a beautiful forest, as you can see here from this satellite or from this drone image. And when you're in this rainforest, you really have the impression you're in the middle of the Amazon, although you're not in the Amazon, but in the middle of this very diverse ecosystem. It's hard to believe that only 100 meters away, we have clear-cut grass occurring. Anelio is a family man. He has three children, and this is the research station he built. So anybody who's interested in visiting the Choco, they have an interest. I don't really know what kind of research people are working on in this research group, but if you have any interest in rainforests in the Choco area of Ecuador, this is an open place. It's all organized. It's a very wonderful place. I spent many days there with Anelio, and it's a great place. There's a lot of, obviously, a lot of incredible species like these two Ananaceae here, but also orchids. We described a new species of Guatteria and Ananaceae from this tropical rainforest. Together with Anelio, we always include Anelio in our scientific outputs, and together with a young Ecuadorian botanist called Nicolas Zapata. And we called it Guatteria esperanzae in honor of this Esperanza Tropical Reserve. Several other new species were described, but there are also a lot of common taxa there. But the real stars of the rainforest are the monkeys that we can find there. In particular, the brown-headed spider monkey, which is a subspecies of this species and which is endangered. Why do I say that? Because this is what attracts funders from the beginning. When we want to protect something, they don't really care about plants, but if you say there's an endangered species of monkey, then money starts coming in. And so another fundamental person in this project, and who basically is leading everything, I just provide scientific support, I would say, is Andrea Narvaez, a biologist from Ecuador, and she has this great leaf foundation. She's a zoologist, and here you can see that we're training Anelio to try and place camera traps to try and document this fauna. The fauna of this very small patch is incredible. We have monkeys, we have tigrillos, we have a huge amount of different types of rodents. We've documented a huge amount of biodiversity in this very small 1 hectare patch. And this is all led by Andrea Narvaez and her Great Leaf Foundation. We also undertake reforestation projects. So Anelio has donated part of his land adjacent to the reserve for this plantation. It's a very hard work, reforestation. It seems easy in the cities, but outside in the wild, it's very hard. A lot of invasive species, so it's a long work. We have a lot of volunteers. Here we have the lady on her knees. There is a volunteer from the village where this rainforest is documented. And here we have my son, Luca, on the left, and one of his friends who went for a week doing an internship trying to help with the different activities of the project. We also have a 1 hectare plot inventory. Again, a very long work, which we undertook together with a master's student that you can see here on the left side looking up, Ignacio Corral, together with other botanists led by Anelio, where we looked at tree density, tree mortality, and we did a lot of phenology and generated a checklist of plants. Again, don't have much time to go into the details, but this is a whole nother fascinating result that we had for this forest. And finally, the most important is environmental education and implicating the local people of the villages around. Here we have, led by Greatleaf, Andrés Narváez, providing activities to inform the younger generation of why these rainforests are important. We also have tree climbing activities led by Nico Tinoco, who's a biologist expert in climbing trees, and he's placed a huge amount of camera traps in the canopy, which also allows us to document all these canopy dwelling species in the forest. I think this is an active collaboration, and we applied for funding. And one of the biggest results that we had was to get funding from Franklinia, which allowed us to buy this rainforest, which is right next to La Esperanza. This forest was up for sale, and anybody could have bought this and shaved or cleared this cut very quickly. So it's always a race against time in these areas to save these patches. And I was particularly very happy because this patch of forest also has the highest the tallest palm in the area. You can see right here on the top left, this palm sticking out, a very, very tall palm. And so we were able to save that palm also in the nick of time. So I'm going to stop here. Maybe I went overboard a bit. I had a lot to say. I just wanted to thank all my collaborators, all my funders. And once again, thanks to Julissa for inviting me to give a talk at your seminar. Thank you.
Okay, thank you. I believe our camera just gonna just give me 1 second. Are there any questions for Dr. Thomas Couvreur in the audience? Yes, 1 second. Hopefully that worked. If not, if anybody has any questions online, please type your questions in chat so I can say it to our audience here as well as to Dr. Thomas Couvreur.
Hi, can you see us? I don't know.
No, I don't.
No,
I don't.
okay. Okay, I'll just.
I can hear you though. That's fine.
Okay, that's good. I was gonna wave, but then okay. Thanks so much for a great talk. So my name is Adam Meyer. I'm a PhD candidate here in the Ecosystem Ecology Lab. I was really interested in some of the points you made about, if I understood you correctly, how you can use macroevolution to test ecological questions like top-down control by herbivory over large scales. So I thought that was just really cool. And I was wondering, since you were talking about herbivory, I was wondering about top-down control by predation, and if there's something that macroevolution can tell you about patterns of, trophic cascade or top-down control of herbivores by predation. And if you haven't thought too much about that, it's pretty different from your talk, that's okay. But just really curious because the idea seemed really cool.
I mean, the part of the project was to test like herbivory, but I have to say that our approach to that isn't ideal, but the challenge was to try and generate a data set at the global level. So obviously we can't do what Paula did across all of the tropics. We wouldn't have time to do that. So what we were doing is using near infrared scanning to try and estimate, and I really want people to remember the estimate. We're not really identifying chemicals in the leaves. We're just trying to estimate what kind of chemical diversity are in these leaves and if they're different between species. And about your question, I'm not really familiar with all these I mean concepts that you can link them to macroevolution. But basically in macroevolution you can test whatever you want, as long as you have the data set to do that, and it's a pattern that occurs at evolutionary timescales and not at more recent or day-to-day timescales, I would say. But I don't have too much more to say about that, even though it's a great question, and I haven't really thought about that, to be honest.
Thank you.
All right, perfect. Are there any other questions in the audience or online?
Hi there. Thank you again for your talk. Amazing. Just like everyone has said, really good images, and I really like the way you laid it out for us. At this earlier on in your talk, you talked about unchecked data from sites like GBIF, and I was wondering, considering this tool that you presented for doing preliminary assessments of species for the IUCN Red List, with social media sites like iNaturalist influencing biodiversity data now, how do you see, like, I guess my fear is that having a tool like this ConR preliminary assessment paired with this availability of huge biodiversity data sets that are unchecked, like you said, do you think there's a risk that the tools are taken up and used really aggressively, but that idea that you said, you know, we do need to check these data, that gets lost kind of in the mix of, you know, we have these amazing tools and we can do this really fast. Do you think there's how do you see that these fast tools and these big data sets getting integrated with actually continuing to study biodiversity and species?
Yeah, that's an excellent question, and something I'm confronted to all the time when I talk to people about this. So, and there are multiple responses. First, it depends on what you want to do. So, if you're a researcher, we imagine this tool to have two main purposes. The first one was to undertake large-scale assessments. Obviously, we're not going to spend 10 years assessing all the 22,000 species of plants in tropical Africa, because we don't have the manpower to do that. We don't have the funding to do that. Doing IUCN Red List assessments is really not, it's important, but it's not really funded. I mean, getting grants just to do that is really hard. So we wanted a tool just to accelerate it, but we wanted our tool to be as close as possible to the IUCN methods. So it's not the first time that people come out with all kinds of different ways to assess threats, but none of these ever really followed IUCN. So in our case, we wanted to be able to do this at large scale, but follow IUCN. We had to make sense for IUCN. Now, obviously, we really put an accent on the preliminary. The other one we wanted to have is to have a tool which could automatically generate all the statistics you need, the mathematical statistics, I would say, the hardcore numbers you need to have to be able to do these assessments. Initially, what we had to do was either use ArcGIS, download a little script, and it would take a long time, or GeoCats Online, which only did one by one. Here, let's say you have 10 species, you can put that in R, and you can generate a table. It gives a suggestion of where it should be, but then you have all the statistic at your hand. So you can just print out the table and then start working species by species and doing the expert assessment. So it helps with that too. Now, obviously, a lot of people have abused of ConR. A lot of people have misunderstood what the tool is for, but that's kind of the risk. Everybody's going to misunderstand tools. And in our publications, we've clearly said that it's not meant to be a put it in and then just take the results without criticism. But it is a good help for large-scale analyses. Obviously, using the word preliminary or possible or potentially, those are all the keywords that need to be stayed in there. So I'm not too afraid. I think it helps, and I think but it's important to continue communicating on this and not saying don't use it, but say use it with these caveats, which are they're not, the output isn't the full assessments, and they always need to be checked and rechecked. On the other hand, we're running out of time to do all this, and I'm leading this large project in palms, 2,500 species, together with many colleagues across my network of collaborations to try and assess all these species following IUCN. The other thing I want to say is not because in a publication somebody says it's critically endangered that it is. It's only once it's on the red list that it's official. So I can say, oh, this is all critically endangered, but if it's not on the red list, companies don't have any obligation to follow it. They can just, oh, it's on the red list, then I don't care. I don't have to follow. It's not endangered for me. It doesn't exist. So ConR is important, but basically it's only the assessments that end up on the red list which are official, and that obviously we have to do the full assessments for them to be there. So on one hand, it is worrying, but on the other hand, it's never going to override the system because there's always a review process before that ends up there by experts.
Thank you. That was a really well thought out response. And I really see a lot of value in this tool. Like you also mentioned, if it's not on the red list, it doesn't exist. That kind of underscored for me the importance of assessing species that aren't necessarily imminently at risk, getting those like least concern species assessed, so we at least know where they're at. And a tool like this, like you said, it makes that kind of lays all the information out for you so that you can spend your expert time reviewing instead of doing math and pulling all that stuff together. So thank you. That's really helpful.
Okay, and I have a question from, I'm going to pronounce your last name wrong, sorry about that. Nicholas Loaiza, asking, have you tested if there is a difference between the chemicals in preserved specimens in herbaria than in live plants?
Yeah, that's an excellent question, and that's always going to be the big doubt. Obviously, the main difference, I haven't done this, some of my colleagues in the US have led this research, where they actually nurse a living leaf, and then they dry it, and then they nurse it every year. So they started that, I think, 3 years ago, and obviously the biggest difference between dried and herbarium preserved is the content, the water content, which disappears. But once it's dried, the nurse has a tendency of not moving all that much or not changing all that much. But of course, what we did was test if you dry the specimen using alcohol before or not. And we found that doesn't really impact. I mean, it's a very small scale study. I wouldn't say it's super significant, but at least we haven't found any signs that that these specimens are, or that these techniques to conserve plants before we dry them have a huge impact. What we did see was if something is silica gel dried. So we also tried to nurse little pieces of leaves that were in silica gel, and then we did see there was an impact, and that's probably due to the fact that all these little pieces of silica kind of drop onto the leaf and mix the or mess up the spectrum that we have. But to go back to this was the potentially high impact, but also very high risk part of the project. It was a lot of money and probably would have ended up with just gibberish. But Lily Bennett, the master's student, was able to show that this kind of had made some kind of sense in her preliminary analyses at the Annonacea family level. I wouldn't go into the details here, but it seems like it's making some kind of sense for the moment.
Perfect, thank you. And is there any other questions in the audience? If not, I have one more question online. Okay, online, sure. So I have a question from Katrina Cruikshanks saying, thank you, Dr. Couvreur, amazing talk. I'm curious about the canopy dwelling species. I study lichens growing in tree canopies in Nova Scotia. Are the communities very different, / have you gotten to make collections from the canopy? From a conservation standpoint, have species grown in the canopy been included in assessments at all?
Yeah, very, very good. I guess the question is related to plants. We don't have a proper assessment of, I would say, epiphytic biodiversity or plants growing in the canopy in rainforests overall. It's a very tedious job. One of my colleagues here in IRD is an orchid botanist, and yeah, to climb up a tree and to go from branch to branch is not only a risky business, but also a long and tedious business. So it's a very long process. And we in the Esperanza project, we haven't documented that biodiversity yet, mainly because we didn't have an expert who was able to do these things. I personally don't do that. I would love to do it, but having a wife and kids, there's some things that I don't permit myself to do anymore. I do a lot of dangerous things, but that I don't do. And so yeah, we don't have too much insight in that. But it's definitely a very hard and long process to do. Probably in the future we'll try and find somebody who can climb up, a botanist to climb up, because the one who climbs up is he works on mammals and things like that. So he doesn't really collect anything. So we're hoping to find somebody to go up there and get a good idea of epiphytic diversity.
All right, and one question here, yes.
Hi, Thomas. This is Julissa speaking.
Hola.
Hola.
I was wondering where you were.
Yeah, I'm sitting on the back of the room.
Okay.
And thank you very much for giving such a great talk. It was very inspiring to our department and to our
Thanks.
students. Yeah, it was nice to see you again after so many years.
Yeah.
Yeah, so my question is, when you talk about the neo and the paleo endemics in Africa, and you saw the distributions of old and new, I guess, endemics in Africa, I wonder, what is the threshold value? Like, what do you call old and what do you call new, neo and paleo? Do you have a specific threshold? Like, is it, you know, for calling something old and something new?
Yeah, that's an excellent question. It's interesting because in the canopy model that we use, this is all phylogeny dependent. So we don't say, okay, anything older than 20 million years is neo, and anything younger than 20 is anything older than 20 is paleo, and anything younger is neo. The threshold is calculated based on the phylogeny. So it's all relative, which kind of makes sense because, as anything in biology, it really depends on the scale you're looking at things. If you're looking at very small scales, something might be very old to the scale of what you're looking at, or very young. So the threshold between paleo and neo is not a fixed entity; it moves as the phylogeny changes. And the same is for the endemism, which is basically an estimate. It's a statistical estimate of how many species or taxa or lineages are endemic to the different pixel sizes that you have. And then it calculates a certain number of statistics, and it gives a probability of, that this is actually a high level of endemicity. That's why in my map, many of these regions weren't labeled as having high levels of endemicity, mainly because, well, there were a lot of widespread species there. But in the East African Rift Valley, a lot of these taxa, and I didn't mention that, but we were actually at the genus level there. So that's kind of another caveat to the story. It's generic level. That's because we didn't have information at the phylogenetic level of these species. So you can't apply it if you don't have a phylogeny at the level you're looking at. But at least at the generic level, that made kind of a lot of sense. And yeah, so that's where the threshold is set. It's phylogeny dependent.
All right, thank you. And if there are no other questions in the audience or online, just give a second in case anybody does have a question. No, I believe that's all. All right. Thank you, Dr. Thomas Couvreur. A very interesting talk given. There will be a link posted on YouTube to your talk. And thank you very much from here in Memorial University.
Okay, thank you for having me. Bye, hasta luego.
Okay, bye.
See you soon, Julissa.